Well, welcome back everyone. Again, this is Michelle. What we're going to do now is move on to biological basics, and I'm going to break this lecture into two, one regarding neurotransmitters and the other regarding medications. So as we know and maybe have discussed in the past, mental health all occurs in the brain. So it's important for us to understand the brain and what's going on inside the brain to understand the medication part of it and what we're trying to treat. So let's begin with some basics. Excuse me. My slides are slow again. So what we're going to discuss is functions of the brain. This should be a review of information that you've learned in the past. But what we're looking at is what the brain is trying to do. So it monitors external world. And this is its main function. The brain receives messages via the peripheral nerves and then interprets these messages to take appropriate action in the body. So for instance, if something is hot and we touch it, the brain takes that message from our hand to the brain, processes it, and then shoots back a message for us to recoil our hand from that hot, whatever it is hot that we're grabbing. So that's how the brain is monitoring our external world. Now, with mental health disorders, sometimes the brain, the stimulant does not come from the external world, but comes internally. So schizophrenia, those people often experience sensations that do not originate outside, but inside their brain by the autonomic nervous system. So for instance, if they're seeing or hearing people, that's an internal stimuli that's processing that message in the brain. So another function of the brain is to control skeletal muscle contractions. It initiates the cells to contract and also direct fine motor control. Psychiatric disease and treatment with psychotropic drugs cause movement disturbances. So, you know, especially with the antipsychotic drugs, and I have put a bunch of videos on Blackboard regarding these effects of this medication. One example is dystonia. So make sure that you do go to Blackboard under your view section and watch the snippets of the side effects of these medications so that you understand what you're looking for in your patients. This is also things that we're measuring with the AIMS screening tool that we discussed previously in the class. So important to remember is that the skeletal muscles are controlled by the brain, and this includes the diaphragm. So it's essential, which is essential for breathing, of course, and the muscles of the throat, the tongue, and the mouth, which are essential for speech. Thus, drugs that affect the brain functioning can stimulate or depress respiration or lead to alterations or abnormalities in speech. So we always, as nurses, need to make sure that we're monitoring respiratory status and swallowing ability. So another function of the brain is our sleep cycle. So unfortunately, many of the drugs that we're going to use to treat psychiatric problems will interfere with the normal regulation of sleep and alertness. The connection between all of these functions of the brain are equal to the results of the actions of the individual neurons and the transmission of information within the brain. So the neuron is a nerve cell that conducts electrical impulses. It has three responsibilities. One is to respond to stimuli, two is to conduct that electrical impulse, and three is to release a neurotransmitter. And the brain is composed of approximately 100 billion neurons. Functions of the brain result from the action of individual neurons and the interconnections between them. The electrical impulse goes from one end of the cell to another. This electrical impulse consists of changes in the membrane permeability that allows the flow of sodium ions and the outward flow of the potassium ions. Once an electrical impulse reaches the end of the neuron, a neurotransmitter is released. So, how does this work? Well, it's how depolarization and repolarization works. So depolarization is the change in the membrane permeability. The sodium moves in and the potassium moves out of the cell. The cell membrane becomes positive on the inside and negative on the outside. And this causes the cell to depolarize. Okay? You can kind of see that in the picture. Repolarization occurs when the cell achieves homeostasis by the movement of potassium out of the cell until the neurotransmitter is released in the synapse. Once depolarization occurs, the next cell is then propagated until the message is sent. So if you look here, watch my mouse. This is your pre-synaptic cell. This is your post-synaptic cell. This is your neurotransmitter. So the neurotransmitter is released by your presynaptic cell and is received by all these receptors on the post-synaptic cell. Okay? This is depicting the exchange of sodium and potassium, which makes the depolarization and the repolarization occur, which takes that electric stimuli or that electric message and transfers it down the neuron to the presynaptic cell. Okay? This area in here between is your synapse. Okay. So the propagation of the electrical impulse from one neuron to another serves as the means of communication between one part of the body and another. So once the electrical impulse reaches the end of the neuron, a neurotransmitter is released. You probably remember this from your readings. A neurotransmitter is a chemical that functions as a neuromessenger. Depending on the neuromessenger and the receptor, the next neuron will either be more or less likely to initiate an electric impulse on the postsynaptic cell. Neurotransmitters are released from the axon terminal at the presynaptic neuron, and this neurotransmitter then diffuses across the space, the synapse, to an adjacent postsynaptic neuron where it is attached to the receptor on the new neuron surface. It is this interaction from one neuron to another by way of neurotransmitter and receptor, that allows the activity of one neuron to influence the activity of the other. Excuse me, I have to take a drink. Okay, neurotransmitters are destroyed via immediate inactivation at the postsynaptic membrane or the reuptake by the presynaptic cell and either recycled or inactivated. Excuse me. So, the neurotransmitter carries the message from one neuron to the next, right? It excites, inhibits, or reuptakes, or reuses, or destroys extra neurotransmitters. So the excite is when it passes the message on. The inhibit is when it stops the message. And the reuptake is when the neurotransmitter is taken back into the original cell. So watch my, my cursor here. So here it is being retaken back up into the presynaptic cell, And then there is a component in here called monoamino oxidase. This right here is responsible for destroying neurotransmitters that we no longer have use for. Now, the four main neurotransmitters are norepinephrine, dopamine, serotonin, and acetylcholine. Okay? So mental health conditions that we deal with, either we have too much of these neurotransmitters or too little. So this slide here depicts the problems that are going on. Okay? So deficiency or excess of neurotransmitters can lead to mental health disorders. And most things are linked to the frontal cortex of the brain, the basal ganglia, and the upper brain stem. The neurotransmitters that have been most consistently linked to mental activity are, as I just mentioned, norepinephrine, dopamine, serotonin, GABA, and glutamate. Excuse me, I said acetylcholine before. It's GABA and glutamate. If you take a look at these pictures above, you can see what's going on. So your first one is normal. There's equal neurotransmitter to receptors. Doesn't really look like it in that picture, but that's what it's supposed to be. If there's a deficiency in neurotransmitters, it means we have less neurotransmitter and then we do have receptors. There's not enough of the neurotransmitter. Deficient receptors is we have more neurotransmitter than we have receptors to use it. Okay? So not enough neurotransmitters produced or released, or not enough receptors to accept the neurotransmitter. Excess of certain neurotransmitters, too many neurotransmitters, is that are being released, then can be used. Excessive responsiveness is that there are too many receptors to use the neurotransmitter. So let's discuss the main neurotransmitters. I'm sorry, I don't know what is happening to me. So the first is dopamine. Dopamine is responsible for fine muscle movement, emotions, thoughts, and decision making. If we have too much dopamine, what we typically end up seeing is mania or schizophrenic type conditions. We have excessive motor movements, excessive emotions, excessive thoughts, and poor decision making. If we have not enough dopamine, what we tend to see is depression. There's too little, right? So your muscle movements are going to be slowed down. You're going to have depressed emotions, slow thoughts, and poor decision making. The next neurotransmitter is norepinephrine. Norepinephrine affects your level of affect and mood. It affects your attention and your arousal, and it stimulates that fight or flight response that we discussed when we talked about stress and crisis. So if we have too much norepinephrine in our system, we're going to see things like mania, schizophrenia, anxiety. If we have not enough norepinephrine, we're going to have conditions like depression. Serotonin is responsible for our sleep regulation, our sense of hunger, our mood states, and our pain reception. Also responsible for our hormonal activity, and it also plays a role in aggression and sexual behavior. So again, if we have too much of serotonin, we're going to have anxiety states and insomnia. Too little, again, depression. So GABA, which is called gamma aminobutyric acid, inhibits or reduces aggression, excitation, and anxiety. It plays a role also in pain reception. It has anticonvulsant and muscle relaxing properties. It may impair cognition and psychomotor functioning. GABA is responsible for slowing things down. The pre and post synaptic exchange is inhibited. It is slowed down. Therefore, that's why with pain, if this message is slowed down, it decreases our perception of pain. One medication that we will see used a lot is gabapentin. It works on the GABA receptors. If we have too much GABA, if we increase the GABA, with the gabapentin, it will slow down the next neuron, and it slows down that excitability. And that's why you get some of that anticonvulsant properties also, because it's slowing down that transmission between the pre-synapse and the post-synapse. So glutamate is an excitatory neurotransmitter, and it plays a role in learning and memory. So in alcohol abuse, glutamate, an excitatory neurotransmitter, actually interplays with the GABA, which is your inhibition neurotransmitter. And there is an interplay there. They function in opposite of each other. Glutamate has been linked to learning and memory, and GABA will slow the glutamate's ability to excite. So if we have increased glutamate, we have increased excitation, and it's very taxing on the body. So therefore, GABA will come in to try and slow down the glutamate from the brain so it's not excited enough, and it inhibits that glutamate, okay? So oftentimes if we have too much glutamate and not enough GABA, what you're going to see is the psychosis kind of symptoms in your mentally ill person. So acetylcholine is another neurotransmitter. It is responsible for learning and memory also. It helps with, it is responsible for mood, mania, sexual aggression. Excuse me. It stimulates the parasympathetic nervous system. And therefore, if we have too much, we can have some depression. Excuse me. And if we don't have enough, you're going to get symptoms like Parkinson's, Alzheimer's, and Huntington's that give you that decreased learning and memory capability. And again, if we have too much acetylcholine, it's going to affect that mood and affect aspect that we could see and have a depressant effect. Histamine is involved in alertness, obviously our inflammatory response, and stimulates gastric secretions. So if we have a lot of histamine in there, it will produce our allergic reactions. And if we have not enough histamine, it's going to have more of a sedating effect and a weight gain associated with it. So these are the main 6 neurotransmitters. The better understanding you have of these neurotransmitters and how the message is sent down the neuron to the presynaptic cell, through the synapse to the postsynaptic cell, the neurotransmitters and how they relate to the receptors, and how if we have too much neurotransmitter, how that can be circled back into the presynaptic cell and either destroyed or reused. So I have posted on Blackboard an overview of all of these neurotransmitters, so it would be a good idea to go back. The book does offer a more in-depth understanding of each of these, but I think what I've provided and the handout you have would be enough information for you to have a good idea of how this stuff works and be able to apply it to the medications that we're going to discuss in the next lecture. So I just wanted to touch briefly on brain imaging. So we have CT and MRI imaging of the brain is really useful if we're trying to look for overall structural abnormalities in the brain that can cause problems. So any large damage or changes in areas are going to be picked up on a CT or an MRI. They do not microscopically look at the different cells, neurons, or receptors. Where we would get that would be from a PET or an SPECT scan, okay? These two have the functionality to be able to actually get right down to the neurotransmitters and see where the problem is with the transmission of these messages in the neurons. And how it does that is we inject a radioactive tag, we call it, into the person. And these compounds are, they attach to the glucose in our system. And as we know, the brain is going to be using up a lot of glucose. So these tags will, carry up to the brain and illuminate the areas that are active in the brain so that we can see where the activity is or is not in the brain. These are some examples of these PET scans showing us activity. So the orangish reddish colors are going to be areas that are very active, and the blue-green areas are going to be areas that are more dormant. So if you look at here, the twins here, you can see a twin who does not have schizophrenia and the other twin who does have schizophrenia, and compare right here, the schizophrenic patient has a lot of activity going on here and the normal patient doesn't. Okay, again, you have some activity here that is missing but is active in the normal. So they use these scans to compare and contrast and try and determine what is going wrong in the brain. Okay, so anti-anxiety drugs, well actually I need to stop there. We're going to talk about drugs separately. So I'm going to stop and the recording here, and you can pick up the drugs in the next one. Okay, thanks.